So way back in November, we talked about virus replication. And a quick recap of what we did here, we had the host cell, the virus particle, and if you recall, the viruses are made up of a protein coat on the outside and then the nucleic acid in the inside, and that can be either DNA or RNA, okay? And it infects, it attaches to a host cell. We have these little projections that attach to the host cell. It then goes in and the virus nucleic acid combines with the host cell nucleic acid, and then we got mitosis. So this is basically, we go through the cell cycle. One cell produces two cells, and it keeps going like this. And so the host cells have now got the viral nucleic acid in. Remember, this is the lysogenic phase, and it's asymptomatic. You do not have the symptoms. Then, at some point, the virus nucleic acid switches on, and it converts the host cell into virus-making particles, which then build up in the host cell, burst out, and go find a new host cell. That's what we did. Now we have done protein synthesis, and now we have done structure of the cell membrane. What we're going to do is we're going to look at this in a little more detail, right? And it looks like this, right? Yeah, it's another one of those crazy diagrams, but remember what I tell you. Follow the arrows. So we start here, right? And I'm going to bring this down just a little bit like that. Right, what we have right here is a virus particle. Let's go ahead and label this virus particle. And you can see this virus is made up of two parts. It's made up of its protein coat around the outside and its nucleic acid on the inside. Now, it's interesting with this virus, you can see that is a single strand. So this would be classified as an RNA virus. So that whole single strand there would be virus. It's an RNA, and since it's all their nucleic acid, it is the virus RNA genome. Okay? And then you've got these, we called these projections or attachments before. We're going to name them something new now. Since they're made out of proteins, their job is still the same, to attach to a host cell. We are going to call these spike proteins. Okay? Now, how does a virus get into a host cell, and why do certain viruses attach to certain host cells? What we have right here, if you recall, if you look carefully, we've got a phospholipid bilayer with proteins running, like there's a channel protein running through, and these little markers on the outside. This, for example, since we are in the middle of a pandemic dealing with COVID, this could be specifically a lung cell. We know that the COVID-19 virus attaches to lung cells. And you'll notice that this little spike protein here fits perfectly with this little marker here in the host cell. And obviously this part here, that is the cell membrane. Okay, that's the cell membrane. Now, once the virus particle has attached to this marker in the cell membrane of a lung cell, the RNA is going to travel to here. Now, I'm sure you're looking at that thinking, I don't remember, I have no clue what that is. But let me take you back to quite a while back to this. And you'll notice here is the nucleus, here is the endoplasmic reticulum, and all these little black dots are ribosomes, which we now know are where translation takes place. Okay? So, the virus RNA has attached itself to a ribosome. So right here, this is the virus RNA, and it's kind of like RNA viruses cut out transcription, because remember, transcription is the DNA opening up to make messenger RNA, which then leaves the nucleus and goes to the ribosome. Well, we've already got RNA, so we don't need to do the transcription part, we just need to do the translation. And right here, these black dots here, these are ribosomes. And these will be right next to, that will be the nucleus. Okay? Oh my goodness, I spelled nucleus wrong. M-E-E-U-S. How can I spell nucleus wrong? Okay, so, and you can see here, this is clearly RNA because it's a single strand. And you'll notice there we got uracil bases. Okay, so what happens in the RNA? Real simple. Here is the ribosome, right? Here is the viral, and I'm going to call it messenger RNA now because it's doing exactly what messenger RNA does, which means that 3 bases here is called a codon. Okay? And these up here are my incomplete transfer RNA molecules. Okay, so if the codon is GUC, the anticodon is going to be CAG. And over here, this codon is UCA. So over here, I'm going to have A, G, and U. Okay? And at the back end of each one, there is our amino acid. Which amino acid is it? Well, I can quickly pull over here and go to my codon chart. Remember, codon chart, so it's the mRNA. So UCA, UCA, that one there is going to be serine. So I'm going to write SER. This one here is GUC. So come down here to G, U, and C, and that amino acid is going to be valine. See, G, U, and then A, valine. Okay? So, what's happening is our ribosomes in our lung cells are using the viral mRNA, the viral RNA, to assemble amino acids together in a specific order. And correct me if I'm wrong, but I'm not. That is translation, isn't it? That is exactly what we've been talking about with protein synthesis. So what we're doing is we're using the viral genome, we're using viral code in our cells to make not our own lung cell proteins, but virus proteins. Now, making proteins is a bit like buying some flat pack furniture. It comes in all these various parts and you have to put them together. So once all these proteins have been assembled, have been produced in the virus, they need to come to this organelle, which is the Golgi body. And again, if you go back and look at that big picture of the cell you've got, you'll see that what happens in the Golgi body is assembly of proteins, right? And what are all these proteins? They belong to viruses, right? So the assembly of the proteins into virus particles. Okay, so we're using our cell mechanisms to make proteins. Those proteins go to the Golgi body, and then all those various parts get put together like flat pack furniture, and we make new virus particles. And just like before, those virus particles build up, and eventually the cells, the lung cells which are producing them, are not going to be able to tolerate all these virus particles in, and they're going to get destroyed. And then what happens is this: you've got all these virus particles in your lungs. If you sneeze, cough, talk, all of those virus particles are going to come out of your respiratory system. They're going to come out of your lungs, up your trachea, out through your mouth, and they are going to go and find new host cells. They're going to go find someone else to infect, right? To infect. So what we've done is we took what we knew already, we added our understanding of the cell membrane, we added our understanding of transcription and translation, and how virus particles are produced. Now, the reason this is so important is because when we get sick, we want to try and prevent ourselves from getting sick. And one of the things we have been hearing a lot about is vaccines. The new vaccines for the COVID-19 virus, they do not inject you with the whole viral genome. They only inject you with a very small part of it, which is designed, it's the little part of the viral RNA, that is designed to produce virus spike proteins. So when we get to we go through the whole process, what we're making are not whole virus particles which can then leave and go and infect someone else. We are only producing this part, the spike protein. And the reason that's important is that if you have spike proteins, they are what we call an antigen. They're produced by the virus, by the pathogen, and they can start an immune response in our bodies to produce antibodies that will recognize spike proteins should a virus particle infect you again. More of that later. I hope that kind of expands a little bit on virus replication, and particularly with relevance to what we're going through right now in the pandemic.